All right, it's 7:32. We'll get started. Good morning, everyone, and welcome to Surgical Grand Rounds. It's my pleasure to introduce Armen Parsyan today, who will be our speaker. Armen is well known to the McGill surgical crowd. He is currently an assistant professor of surgery, oncology, and anatomy and cell biology at Western University in London, Ontario. He's a breast cancer surgical oncologist at the London Health Sciences Centre, where he also leads the breast cancer research laboratory at the Verspeten Family Cancer Centre. He's also a breast surgical oncologist at St. Joseph's Healthcare in London. Armen has had a very rigorous, very international training, I would say. He completed his medical degree and surgical training in Armenia, after which he moved to the US, where he completed a master's in public health and epidemiology in Boston at Harvard University. From there on, he moved to a third continent. He went to England, where he completed a PhD degree at the University of Cambridge in molecular virology and immunology, and then finally came to McGill initially for his postdoc under the leadership of Dr. Naim Sonnenberg, after which he completed a residency in general surgery, moved on to Toronto for a breast surgical oncology fellowship, where he also got to work with Dr. Takmak at Princess Margaret Cancer Center and is now staff at Western. Armen is a very busy academic surgeon. In addition to his practice, he's received numerous grants, competitive peer funding agencies, as well as awards. He's published in in some of the most prestigious journals, Nature, Science, PNAS, and others. His research focuses on the utilization of modern translational breast cancer models and technologies to study mechanisms of resistance and sensitivity to novel and existing chemotherapeutic agents, to identify biomarkers and drug targets, and to better understand the pathogenesis of breast cancer. He'll be talking today about identifying novel molecular vulnerabilities in triple-negative breast cancer. Armen, welcome back to McGill.
Well, thank you very much. Glad to be back, and thanks for the invitation. And I want to thank everybody for inviting and organizing this. So I'm going to talk today about one of the pipelines within my lab. It might be a little bit more molecular specialized, but I hope you won't find it too boring or too technical. So we're going to talk today about identifying novel vulnerabilities in triple-negative breast cancer, and specifically on an example of a protein called Artemis. So as Dr. Magardichan mentioned, I'm a clinician scientist at Western University and London Health Sciences Centre, where I work as a breast surgeon, but also lead the breast cancer laboratory. So, I'll see, yeah. So I declare no conflicts of interests, and I'll give you some introduction about breast cancer just to refresh some who are not in this field. So breast cancer is the most commonly diagnosed cancer in women worldwide, and in Canada in particular. 1 out of 8 women are diagnosed with breast cancer during their lifetime. And it's the second cause of cancer-related mortality in women as well. So it poses significant burden, not only individual, societal, but also on a more broader scale, economic and others. So breast cancer is not a single disease. It's very heterogeneous, and there are several attempts to characterize its heterogeneity, which eventually lead to kind of the most commonly used and simplified version of it when you divide breast cancer into different subtypes, mostly based on estrogen, progesterone, and HER2 receptor expression. And when neither of these receptors is expressed, then we classify the subtype as a triple-negative breast cancer, although there are some other classifications. For simplicity, I'll just go with this one. So this is prevalent within overall breast cancer package. 1 out of 7 women approximately are diagnosed with TNBC, and approximately 15% of all breast cancers. Now, what is particular about TNBC or triple negative breast cancer? That is generally considered the most aggressive and lethal subtype. It's quite often treatment resistant, and due to the lack of these receptors or biomarkers, it lacks targeted treatments, although there are new developments, and I'll mention that as well. So main treatment strategies for breast cancer involve surgical, if it's not distantly metastatic, chemotherapeutic agents, anti-hormonal treatments, not for TNBC, but for hormone-positive ones, targeted therapies in TNBC in particular, for example, PARP inhibitors are used. There are also some antibody drug conjugates like TROP2 inhibitors that recently being developed, etc. So there's a progress in that direction. Immunotherapy as well is now also used for triple negative breast cancer, and another cornerstone treatment is radiotherapy. So with all these modalities, however, we strive to achieve better outcomes in our patients, especially with TNBC. As I said, it's quite generally aggressive disease. So what are the clinical needs in this disease is probably the two most important is to identify new biomarkers and also to develop new treatments, either based on these biomarkers or otherwise, and to develop precision oncology strategies, which is essentially the individualized approach to treatment, which is particularly important for diseases with high heterogeneity. And TNBC by itself, it's also very heterogeneous, some classified to more 6 subtypes within TNBC. So that understanding of individual variability is important to have more precise treatment approaches at precision personalized medicine. Understanding pathogenesis of the disease is very important for all of this, as well as understanding treatment resistance. So I will talk about this treatment resistant part, although this talk touches biomarker development and new treatment developments as well. So there are several projects going on in my lab in terms of identifying these new vulnerabilities. And vulnerabilities, what I mean by that, it's some sort of markers that can be used again for prognostication, for patient stratification, for treatment that can be targeted. So one of the projects that I'm working on is identifying these vulnerabilities using spatial omics techniques, which is a relatively recent, very interesting technology, including spatial transcriptomics and proteomics. And I also investigate various novel drugs using these vulnerabilities, such as CFI 945, which targets polo-like kinase 4. And also I studied the mechanism of resistance, treatment resistance, and particularly to radiotherapy. So within that latter sort of package, we aim to identify a potential targets or again molecular vulnerabilities that might interfere with the response to radiation, or like simply say like radio genes that might be associated with radio sensitivity or radio resistance. So in here, I think one of the learning objectives, if there is such here, is that to basically give you outline how potentially these vulnerabilities can be discovered. So what we did basically, we used the CRISPR-Cas9 approach. So CRISPR-Cas9 is a gene editing technology that can knock out genes, can overexpress genes, can activate genes, and now it's very commonly used technology. We kind of went one step further from CRISPR-Cas9 and used genome-wide CRISPR-Cas9. So what it means that we use a library of single guide RNAs, or it's sgRNAs, that contains this sgRNAs against all protein coding genes within the human genome. So it's the library that we used called Moffat Toronto Knockout CRISPR library, second version. So it contains triplicates or fourplicates of the HGRNAs against each individual gene. That is protein coding. And we use as a system MDA MB231 TMBC cells. So how this works is that we create lentiviral particles that each of them contain one of these HGRNAs, and then we infect the cells, which leads to the transduction of this CRISPR-Cas9 system into the cell and alteration of the gene expression. However, when you do it on a genome-wide scale, basically it's kind of very technical experiment that you infect the cells such so multiplicity of infection with specific multiplicity of infection that each cell gets one HGRNA. So each cell technically or ideally would have a knockout on only one gene. And so using that approach, we created a colony. As you can see here, we infected cells with these lentiviral particles with HGRNAs with anticipation that each individual cell within that plate or that colony that we are using has one gene knocked out. And so for 18,000 genes, 3 times or 4 times, because each HGRNA for each gene is represented by 3 or 4. So then we grow this colony, and then there will be like mixture of cells that some contain knockout from gene A, another will have gene B knockout, etc. And then we split them randomly after they expanded and subject part of them to 4 gray of radiation. And then we let this grow. And when it grows, we waited for 25 days and then collected the cells and essentially extracted the genomic material and then sequenced these HGRNAs, which have specific barcodes representing each specific gene that they knocking out. And then using that approach, we can see the difference between non-irradiated and radiated cells in terms of the representation of these HGRNAs. The HGRNAs that dropped or absent from the irradiated cells, those would likely be represented by ones that participate in the radiation response. So this is called dropout CRISPR screen. And by comparing this HGRNA presence in control and radiated cells and using bioinformatic analysis, we can identify the list of genes that, if knocked out, would lead to the radiosensitivity. And on contrary, if they overexpress, one would expect it will lead to radio resistance. So using that approach, we wanted to identify which genes in TMBC participate or which proteins participate in the response to treatment, to radiation treatment. So basically, we counted the cell growth, and you can see that the radiated cell grew slower as expected because they are radiated and because they have some genes that are potentially knocked out with radiation would grow slowly. So this is expected kind of control follow-up on cell growth. And then after we did bioinformatic analysis, we identified 172 radiosensitizing genes, and we excluded survival-related genes because if they are knocked down, and cell would die and drop out from the screen. But we don't want to contaminate our screen by survival genes. We want to only focus on ones that are not related to survival to that extent, but lead to the radiosensitization if they are depleted. So that's probably the most difficult part to explain here for me, but essentially that's the CRISPR-Cas9 screen that we performed. So what we identified through this bioinformatic analysis of this dropout genes that we found on radiation versus non-radiated control condition. So we found that there are enrichment of certain pathways, meaning that several pathways that contain various genes that do the same function might be overrepresented upon knockout of these genes and with radiation treatment. And one of the top pathways that we identify using KEGG pathway analysis was non-homologous end joining. And this is the pathway that is one of the canonical pathway together with homologous recombination that participates in DNA damage repair. So when DNA is damaged, these pathways get activated to repair this damage. And as we know that radiation, one of the main mechanisms is damage to the DNA. So this makes sense what we observed. And then we looked at the individual genes and saw the scores, like at what extent they are important, quote unquote, in the response to radiation. And using again bioinformatic techniques and false discovery rate less than 0.005, we identified several genes here. I don't go into details of that, but the top gene that we identify was this one, DCLRE1C or Artemis. That was the top hit, which is a part of NHJ pathway too. So things started to make more sense, kind of coalescing together in a logical fashion. Now, what is this Artemis protein? We started to dig into that more. So this is encoded by this gene, DCLRE1C, which is DNA cross-link repair 1C. It's a component of an HEJ pathway or that DNA repair pathway, and it's an endonuclease, meaning it kind of trims the DNA. It plays an important role on VDJ recombination that you know is a key component for creating immune cell diversity and immune repertoire diversity. So interestingly enough, when this protein is malfunctioning and there are mutations that lead to that are inherited, it leads to SCID, so severe combined immunodeficiency, but with some nuance here, with increased radiosensitivity of the cells from patients with this SCID. So causes SCID RS, that is very rare condition that and especially prevalent in some Native American populations such as Navarro. So quite interesting. So we continue digging into it, and especially understanding that it's not much known about this protein in the sense of cancer and mechanism of cancer in general, not only in TNBC. So just a brief sort of summary here from one of our reviews that we published on Artemis in cancer. So the role of Artemis in this non-homologous end joining is essentially what happens that when DNA is being cleaved due to some external factors such as radiation or some DNA damaging chemotherapeutic agents or spontaneously or for some other reasons, there are a set of signaling cascades that are being activated and that involves different proteins that bind to this DNA and then eventually recruit Artemis. And what Artemis does, it trims this ends of the broken DNA. So then ligase 4 can join this end, so then to repair the DNA damage. So this is the function of Artemis and HEJ. In VDJ recombination, what happens that our own cells, immune cells, in this case, they have a RUG complex that intentionally cleaves at the sites of the VDJ region and creates this double-stranded breaks with hairpins. And Artemis here comes and cleans these hairpins. So then subsequently VJ joining can happen. And that, depending on how it cleaved it and how the joining happened, it would create the diversity of the immune repertoire, specifically antigen receptor. So it's quite important protein, Artemis. Aside from this function, it also, we saw that it's, I mean, we looked into the literature, it also participates in other cellular functions such as antagonizes homologous recombination, participates in cellular homeostasis, protein degradation, chromosomal stability, as well as regulation of the cell cycle. So we then went to the kind of next step. So that is it worth examining this protein? And what we did, we looked into the public omics databases, one of them being TCGA, Cancer Genome Atlas database. where we looked at the expression of Artemis mRNA in normal breast and various breast cancer subtypes. There was not enough data on protein expression, so we looked at mRNA. So, and what we found was quite interesting. We found that Artemis mRNA is actually overexpressed significantly compared to normal breast tissue or compared to other breast cancer subtypes. So that's interesting. Then it may be participating specifically in TNBC pathogenesis. We also queried whether this increases in transcript levels of Artemis have any meaningful correlation with the patient outcomes. So we used GPA analysis, and what we found that high Artemis expression, mRNA expression, actually correlates with a poorer survival and significantly poorer survival in patients with TNBC. So that basically tells us that number one, we identified this protein that participates in radiation, modulating radiation response. Physiologically, it makes sense what we found because this protein is part of an HEJ that repairs DNA, that is part of the effect of radiation. And then also through the in silico analysis, we found quite interesting data that it might be overexpressed in TNBC specifically and be correlated with patient survival. So then now we have to validate what we have from this CRISPR-Cas9. It's a big, messy experiment and there might be no specific findings. So to validate this finding that Artemis downregulation leads to the increased radiosensitivity, then we had to create individual CRISPR-Cas9 knockout cells, knockout for Artemis. So we use individual sgRNAs for Artemis and four of them, and you can see here in two different TNBC cell lines, and some of them achieved like a complete knockout of the protein in this line. So then we created a system basically that we can use together with control Artemis expressing and Artemis knockout cells and see what functional implications the Artemis knockout can have in TNBC. So we went with functional experiments. So the functional experiments that we did, we started with colony formation assays, which is a gold standard for assessing effects of radiation on cancer cell proliferation. It's essentially putting cells in like plating cells in a format that a single cell and then after grows the colony, and that colony afterwards counted, and you compare between different treatment conditions whether the number of colonies changed or not. So as you can see here, with control, meaning that Artemis is intact, but it has like scramble sgRNA, to have the proper control for CRISPR system. We then see that with radiation, with increasing doses of radiation, it's not that clearly seen maybe, but you can see there are less colonies, less dots here as we increase the radiation dose. Interestingly enough, and this is quite new finding, that we also observed that if we knock out Artemis, so this is like two different cell lines with like same cell line, but with different sgRNA knockout, we can see that when we knock out Artemis, there is a decrease in colony formation, so in proliferation. And you can see it from this graph that you can see with Artemis knockout, it's almost 50% decrease in the proliferation of the cells that we observed. Moreover, when we combine this with radiation, then we observe effects that basically there are almost no colonies observed. So it appears as if Artemis knockout with radiation then increases anti-cancer effect, and Artemis depletion sensitizes cells to radiation. And we confirmed it in different cell lines, cell models, including 231, MDA, MB231, TNBC line, and SUM 159. Okay, so basically this is a new finding that this protein not only participates in radiation response, but also it's important for proliferation of TNBC. Now, the next step was to see if that we can reproduce that, or what happens if we do the same, but in the in vivo system, in mouse models. So the experiment here is that we we create xenograft models with not-skid mice by injecting these different types of cells, one containing Artemis, others not containing it, and radiating or not radiating them. So we have four conditions here. And then we wait and see how that xenograft grows under different conditions. And so what we observed here was also quite interesting because what we see that when Artemis is knocked out alone, the survival or humane endpoint survival, and I'll explain what that means, increases. So mice kind of live longer if there is no Artemis in the xenograft. So what that humane endpoint survival is that we grow this xenograft in a mouse and wait to the certain point. When it reaches certain volume, we have to euthanize the mouse because of the ethical implications, right? So that's called humane endpoint for the assessment of the mouse survival. And so it's called humane endpoint survival. It kind of approximates overall survival in clinical patients, I think. So you can see that when Artemis is gone, then humane endpoint survival is extended quite significantly. Furthermore, and radiation itself also increases survival, which is known, it's like a known treatment. And then when you combine both of these modalities, so Artemis knockout with radiation, which is this purple graph, we see even further increase in the survival. So mice live longer to the point that some of them live like, I don't know, almost 6 months. I think a couple of them we euthanized because like experiment had to stop. So they were still alive. So essentially what this shows that Artemis participates not only in the proliferation, but tumor growth, as well as humane endpoint survival of mice, and its knockout improves these parameters, while its combination of this knockout with radiation further increases survival and slows down the growth of the cancer cells. So then the next step, we wanted to see what is the mechanism, like what happens when you knock out Artemis and when you radiate the cells together. So for that, we use basically transcriptomics approach, like a bulk transcriptomics, RNA sequencing, where in these four different conditions under no treatment and with intact Artemis knockout, number two condition, number three condition, Artemis intact plus radiation, number four condition, Artemis knockout plus radiation. We took the genomic material, we took the transcripts out, we extracted mRNA, and then sequenced it, and then did bioinformatic analysis to see what genes and pathways are changed upon the knockout of this protein. And there are several, but overall theme was that there is seems to be activation of anti-cancer phenotypes such as immune regulatory mechanism. For example, signaling by interleukins, toll-like receptors, which participate in innate immunity. The font is a little bit small, sorry, but that's essentially what we found. We also found certain genes that participate in senescence-associated phenotype, with senescence being one of the key mechanisms for like in oncology. And what it does, it stops the cell division. So in oncology, it basically stops the division of the cancer cell. So we found several proteins that were overrepresented or overexpressed in Artemis knockout cell compared to control, including interleukin-6 that I will talk more about. And this ID2, for example, it was down-regulated, and this is a negative regulator of these senescence-associated phenotypes. So again, to conclude, we saw like immune regulatory pathways being altered upon Artemis knockout, and also some other pathways, including senescence. When we compared the Artemis knockout with radiation to control cells, then we saw again changes in the immune pathway regulation, such as signaling by interleukins, but also we saw this senescence-associated secretory phenotype pathways being changed. So this was more pronounced in this combination condition when Artemis is knocked out and cells are radiated. So, plus we also found some pathways that are related to DNA damage response, and this is understandable. So then, understandable because of the function of Artemis and also because of the mechanism of radiation treatment. So then we continued our analysis of these functional roles, what happens after we knock out the protein and radiate the cells. And to be kind of brief here, we found that this combination leads to the cell cycle arrest, which is also might contribute to senescence because cells are arrested, they don't grow, and it might activate senescence mechanisms. And we can see here that in, again, different TMBC lines, there was on combination, combination conditions, we see increased of cell division arrest in G2M phase of the cell cycle. So apparently these treatments lead to the modulation or changes in cell cycle. And then we wanted to see if these senescence-associated phenotypes that we are observing, are they real or no? It's like another bioinformatics fluke. So we performed functional experiments with our cell lines where we assessed for the senescence using beta-galactosidase staining, which is this blue stain. You can see the cells that are stained like blue like this. It means they are in senescence, so they stop dividing. And that's good for cancer treatment. You want cells either die or stop dividing. So what we observed here, so in some cell lines like 231, we see that Artemis knockout leads to the senescence phenotype. But when we combine Artemis knockout with radiation, there is a pronounced increase in the number of senescent cells and significant compared to other conditions. And so the same was confirmed in another cell line. So basically the message here that Artemis knockout with or without radiation leads to the dysregulation of the cell cycle, likely deregulation of immune pathways within the cancer cell, as well as augments basically senescent phenotype and leads to the senescence phenotype. So then we wanted to join this together, like what is causing it, like what pathway, because there are several signaling cascades that might do that. One of the pathways that we saw, and I mentioned interleukins here, was interleukin IL-6 JAK-STAT pathway. That is a critical pathway for activation of senescence or senescence-associated phenotype. And what I mean by senescence-associated phenotype is that when cell starts to senesce, it starts to produce cytokines, proteins, different things that create like this signaling microenvironment within the tumor that tells the tumor to stop growing. So this JAK-STAT pathway is one of the critical pathways. We wanted to see if it is altered basically when we knock out Artemis and radiate the cells. And we actually hit a jackpot here by seeing that if we knock out Artemis and radiate cells, we have a significant increase in interleukin expression in different cell types. And then its downstream target of that cascade, STAT3, which starts to become active when it phosphorylates, so phosphostate or PSTAT3, as you can see. We can see there is an increased phosphorylation of for STAT3, while its expression remains more or less the same. So this indicates that this alteration of decrease in Artemis expression or depletion of Artemis and radiation likely leads to this senescence phenotypes through activation of this JAK-STAT pathway, which is another sort of vulnerability can be explored. I mean, this pathway is very well known. We're just linking it to what we are observing. Now, then the next logical question comes. So I just conclude here, basically using all this fishing expedition with genome-wide screen to identify targets that are important for modulation of radiation response. We identified this Artemis protein as one of the top targets and non-homologous end joining component. And then through our individual knockout experiments, we observed that Artemis depletion or Artemis is an important component of TMBC proliferation, senescence, cell cycle, and its depletion together with radiation activates these pathways, including JAK-STAT pathway and potentially other pathway, including immune regulatory ones. So with that, it looks like this protein is important for the pathogenesis and treatment response in breast cancer, and as such can be considered as a vulnerability. So what we can do with that vulnerability? So now we started to look into, are there any inhibitors? Are there any drugs that we can see inhibit Artemis? And so we can basically use them clinically because we cannot do genome editing clinically. So, and we looked into the current literature, including into the electron microscopy, cryomicroscopy images of the Artemis structure. And there were two papers actually that looked into the structure and what they describe. It has several domains, but most interestingly for us and for clinical perspective, the Artemis had this beta-lactam type of domain or MBL fold. And we know that beta-lactams has something to do with antibiotics, right? Bacteria has this. So these researchers looked into antibiotics, certain antibiotics, ceftriaxone and ampicillin, that can inhibit this beta-lactam to see if they can inhibit Artemis actually. But they didn't do that in vivo. So what they did ex vivo experiment. Basically, it's a nuclease assay when you can basically sort of have a biochemical mix of things and then assess if Artemis endonuclease or DNA cleaving activity is maintained or no with these drugs. And they saw using ceftriaxone and ampicillin that they can inhibit Artemis endonuclease activity. Another thing that Artemis also has this zinc finger domains, and these zinc fingers can be competitively inhibited by gold and selenium compounds, such as auranofin and epsilon, which are also approved drugs for, I don't know, rheumatoid arthritis and things like that, but not for cancer. Although some of these drugs were studied in cancer. So they also found that these drugs might be inhibiting Artemis activity. So then we said, and there is no compounds, no new drugs, no new developments in terms of Artemis inhibitors or compounds. So we started with this, so to see if we can repurpose these drugs. So our initial steps was to look at ceftriaxone and auranofin and ampicillin. So we treated the cells with ceftriaxone. And you can see here, it's kind of loaded graph here, but essentially what you can see that with increased dose of ceftriaxone, you can see the anti-cancer effects in TMBC of this antibiotic. So this is not like absolutely new finding because there were some studies, I believe, in lung cancer that they show that ceftriaxone might be inhibiting cancer activity. And we do observe that with increased dosage of the ceftriaxone. Moreover, with the radiation and ceftriaxone, we observed synergistic effects when ceftriaxone sort of increased the sensitivity of the cells to radiation. Just a simple antibiotic, right? That potentially, if this works, can be used for cancer treatment. So the similar results were observed with other cell line, TMBC line, and with auranofin, with this another compound, ampicillin, we couldn't see such effect, so it wasn't working in that regard. So now we are validating these findings because we don't know these effects are specifically related to Artemis or not, so we are doing experiments within that context. So we were persistent, however, and wanted to see like there's no way there is no compounds that might like they are developed somewhere and might inhibit Artemis. So we came across a paper which from quite renowned laboratory in University of Oxford, Dr. Schofield, it's a fellow of Royal Society of UK, like very prominent kind of researcher. And what they did, they were looking into SNM endonucleases, doing a compound screen for different compounds, see if these compounds can inhibit this SNM endonucleases. And Artemis is one of the SNM endonucleases that has three groups, basically, 1A, B, C. Artemis is SNM1C. They were focusing on SNM1A, which I believe is Apollo. They wanted to see if these compounds inhibit Apollo and used Artemis as a control in their, again, cell-free nuclease assays. And interestingly, their whole paper goes on about this SNM1A that they find different compounds that can inhibit it. But then also, like as a footnote paragraph, they also say, oh, look, like this, there are like several compounds that specifically inhibit Artemis. But since that's not of their interest, they basically focus on that. And even Google searches wouldn't find these articles, but we lucky came across it. So now we are looking into these drugs with this Oxford team to see if we can see in vivo effects. So if we can, then potentially they can be translated, like in vivo studies can be done, safety and tolerability and potential translation of this compounds if they work to the clinical practice. So in conclusion, it looks like Artemis is a promising biomarker, and this is based on our finding from in silico or online databases that we can see Artemis expression, mRNA expression correlates with patient responses, patient survival. But it also might be a promising drug target. But why? Because we see that downregulation of Artemis leads to decreased proliferation. So if we inhibit it, it might lead to the decrease of cancer growth. It also increased mouse endpoint survival. So then again, it might have effect on growth and potential progression of the disease, right? And we characterize the mechanisms through which this is working, which is important. So then we can tackle these mechanisms that are activated upon DNA damage response and also can represent a vulnerability in a sense for developing combination treatment approaches in the future. So yeah, and then we identified some non-oncology drugs that are effective against TNBC cells and might be able to sensitize them to radiation. And we are still studying, continue like studies in that direction. So our future direction, actually, my student today is like looking, assessing the protein expression with the pathologist in patient samples that we created tissue microarray with 50 patients. So in this tissue microarray from their fresh frozen paraffin, fresh formalin fixed paraffin embedded tissues, we do the immunohistochemistry analysis and score for Artemis expression, and then we would be able to correlate this with the patient outcomes in our population. So we are working in that direction. So basically, we are advancing our studies towards assessing Artemis as a biomarker using patient samples. We also continuing our study with these non-oncology drugs, such as ceftriaxone, aranofin, disulfiram, and also testing these new compounds from our collaborators in the UK and using different models. I'm not going to go into the details of that. And we also are assessing, since we see that Artemis does modulate response to radiation, which acts, as I mentioned several times, through the DNA damage. So our hypothesis is why it wouldn't modulate the response to chemo, which most of the chemo in breast cancer we are using has one way or another DNA damaging properties. So if that's the case, if chemo damages DNA and there's no Artemis, it cannot recover it, the cell will be overwhelmed genomically and not survive. So we're looking into Artemis knockouts and inhibition in combination with chemotherapeutic agents. And we are continuing our studies in future for the pathogenesis of TNBC. So this study, well, it's a few years now, we were doing it, published recently, and was funded through various sources, including, I don't know, Breast Cancer Canada, CIHR, also this is one of the big medical kind of research granting agencies in southwestern Ontario. So yeah, that's quite. This is my team that worked on this project, including Dr. Allen, who's a vice chair here in research, and Dr. Seskon, who's like growing prominent clinician scientist and medical oncologist, University of Toronto, and Dr. Bhatt, the postdoc fellow that did most of the work on this project. I just briefly mentioned some other projects just to give a flavor what else we are doing. I mentioned this spatial omics, which is like my passion for several years. This technology is, I'm sure you're familiar with bulk omics or single cell omics. This is the next step and actually received like recognition as one of the most advanced technologies or methodologies in Nature Medicine a few years ago was published about that. So what is that technology is that you can assess the expression of proteins or transcript on a genome-wide level, so like transcriptomics or proteomics. But at the same time, you can map it into on the cell, on the tissue. So you can see which cells contain which protein or transcript, where it is downregulated, et cetera, et cetera. So together with the single omics and bulk omics, it gives an advantage also of a spatial mapping of where the changes are, which is critical for assessing, for example, the role of immune microenvironment. This is, I don't know, 5 years of me trying to bring this technology here, and we are almost done. In one of these technologies, we recently submitted paper to the European Journal of Cancer with a very interesting finding here. We found the neurofibromin, which you probably know, like if mutated hereditarily, causes neurofibromatosis type 1, which is a tumor predisposition syndrome. However, not studied in breast cancer very well. And we find that neurofibromin might be participating actually with RAS-MEK pathway in the triple negative breast cancer. Again, I'm not going to go into this much, but again, another vulnerability identified through other approaches such as spatial omics. Another big project that have been working with, now we're kind of slow down on this more, is the polo-like kinase 4 inhibitor that's important for central duplication. And we published several papers regarding this protein and its inhibition, as well as its role in combination with radiation. And just to let you know, this drug is now in clinical trials for advanced cancer, including breast cancer. So yeah, this is this pipeline of mine in terms of assessing vulnerabilities. I guess I'll stop here. And I want to thank you again, and I welcome questions, please.
Thank you very much, Armen. It's impressive to see how far our trainees and our graduates go in advancing their academic careers. We have time for a few questions. I see Dr. Chervenkov's hand. Jean, go ahead.
This was a very, very nice and provocative talk. I'm going to ask you, however, focusing on the cancer, mutation, cell, etc., DNA repair, kind of does not address the big elephant in the room, which is the tumor microenvironment and how the more you hit the cancer, the more you create more EV shedding, more dams, which then go and induce the bone marrow to become even more immunosuppressive by activating emergency granulopoiesis. And so, again, unless we combine these therapies with some way to get rid of the myelopoiesis or reduce it significantly, and again, the T cells will probably be affected by removing the VJ, etc. So, do you plan to combine this with something that hits emergency granulopoiesis? And if not, I would like to offer some collaboration because that's what we do. As a transplant surgeon, that's what I have focused on. And now we're showing some very, very good results along with my oncology friends in thoracic surgery and other parts, namely triple negative breast cancer.
Thank you, Dr. Chervenkov. It's always pleasure to work with you and collaborate. So yes, so I mean, I think if I'm understanding it correctly, there are two parts of that I can look at it as two parts of the question. One is that immune microenvironment locally, and second, like it's a general effect on immune system, right? I don't know what Artemis like inhibitors that we are using, so I start with the second part. I don't know what these inhibitors will be doing in terms of generalized, like changing VDJ recombination. I'm not sure like it will have effect, but I mean many chemo drugs and they cause suppression of a bone marrow. So combining that with something that can boost granular poiesis is a great idea. So I don't think we are there yet because we first need to test these compounds that we have and then actually do like safety and tolerability studies in mouse models before we go to humans even. And so we are in that process, but definitely this is something that I also ask myself, like if you inhibit like this type of protein, what will happen in general with the immune system? On the first part, like immune microenvironment, if like I'm understanding the question correctly, yes, absolutely. I mean, and you can see I was also trying to emphasize the role of these immune cytokines that cancer cell produces and stuff. And all these models are that we see on the Artemis knockout and Artemis knockout with radiation. And the models we are having currently, they are not, they don't have immune system there, right? So, but we are interested, like especially given this IL-6 JAK-STAT pathway, which also modulate immunity. One would be a local immune response. We are trying to see what we can do in like syngeneic models for T1 mice, for example, to assess like what happens with the immune microenvironment. And finally, like again, giving importance of immune system, that's why the different approaches to identifying this vulnerability is important. I mentioned this spatial proteomics kind of briefly study that we are doing, and that was completely based actually on tumor infiltrating lymphocytes. We defined the zones in TMBC, then like did this specific sequencing from these zones, and then et cetera, et cetera. So we identified this neurofibromin actually which is a tumor suppressor, which is downregulated when there is an active immune response in the or T infiltration in TMBC. So yeah, all in all, yeah. And once I have a little opportunity to collaborate with you, I will be first one to reach you.
Yeah.
Thank you.
Armen, actually inducing granulopoiesis paradoxically is bad for cancer. It actually promotes resistance to about everything, including chemo, including radiation, including immunotherapy. And so every time we hit cancer and we think we have gone over the next hump of a mutational, actionable mutation, precision medicine, and if we don't address the suppression of T cells by the emergency granulopoiesis, which ironically push neutrophils and macrophages that are immature and surround even more the cancer, and it becomes a desert of T cells. It just doesn't allow them to come in no matter what you do. That's why CAR T cells did not progress. That's why vaccines did not progress. That's why antigen-specific T cells cannot progress, because you're not taking care of the emergency granulopoiesis and the neutrophils and macrophages. In fact, NLR, neutrophil to lymphocyte ratio, is a predictor of poor response to anything you throw at the cancer. And I don't see anything out there remotely addressing that. No matter what I look, I'm a transplant surgeon, so I did not go into oncology until about recently, but every time I hear another actionable mutation, I cringe and I say, whoops, another one that's probably not going to work and might push the survival a few months, but it's not going to change the outcome. So until we address that elephant in the room, and we have at McGill in my lab with Jonathan Cools, we have shown now in 3 cancers that we can reverse this with our compound. Until we address that, we will not make progress in cancer therapy in a meaningful way to treat metastatic disease. So
Yeah, I mean,
I
I
offer
agree.
my collaboration to your lab,
Perfect.
like I did
Thank
to
you.
Jonathan Cools.
Thank you. So you have a compound basically that sort
Yeah,
of activates this.
yeah, we do.
Yeah,
Yeah,
sounds great. Yeah,
that's right.
that's
We
that's
should
something. Yeah,
celebrate.
We
thank
have,
you so much. Yeah,
we have time
absolutely.
for one last quick question
Thank you.
from Dr. Basak at the Jewish.
Hi, Dr. Parsyan. Very nice talk. And well, I had 2 questions, but I guess I'll have only one, thanks to Ari. And I'm much more optimistic than Dr. Chervenkov is, and I suggest you continue your work.
Armen,
Anyway,
Armen, if you don't mind putting your email in the chat so that those that have additional questions know how to get in touch with you.
Yeah,
yeah,
understood.
Armen, have you considered combining it, not necessarily with chemotherapy, but doing synthetic lethality in DNA repair, i.e., knocking down Artemis either together with other DNA repair agents or in contexts that are DNA repair deficient, such as MMR or BRCA1/2 deficient models?
Yeah, we are not there. I mean, we probably should like look into that more, like this synthetic lethality, like from what I'm understanding, you're suggesting like second gene hit, like hit with second
Yeah.
gene, like in. I mean, we can do that. The thing is that I mean, I'm not sure like this, like it will it be like a lot of genetic manipulation with these cells. Already CRISPR has some kind of non-specific things going in terms of
Well, you have
DNA.
drugs, right? You have drugs.
Yeah, but drugs, yeah, but now
You have PARP inhibitors, for instance, etc.
yeah, so drugs kind of sort of synthetic lethality, maybe like that approach that I mentioned. We will use these chemotherapies, right? Like that are DNA damaging in a Artemis knockout situation to see if Artemis knockout would increase the efficiency of these medications. But in terms of the backgrounds, yeah, I mean, we are looking into this with basically in terms of different phenotypes, right? Of the, I don't know, like BRCA mutated cell lines or MMR deficient, etc. I mean, we are not there yet, but this is something in our, actually I apply for the grants that would like kind of give me more options, including that, but also including the like immune system, right? So yeah, so it's a great question. I think it's a very interesting direction also to take.
Nice talk.
All right. Thank you very much, Dr. Parsyan, on behalf of everyone in the Department of Surgery at McGill. I remind everyone that the chat has the link for your CME credits and attendance record. Have a good day, everyone. Thank you, Armen.
Thank you so much, everybody, and have a great day. Thank you, Dr. McCurdy-Chan.